Hello, everybody. Thank you for joining us. It is my great pleasure to kick off this webinar with Professor Huganir. I'm going to read his bio briefly. I am certain there is a much longer bio out there, and I apologize for all of the things I am skipping. But briefly, Dr. Huganir is a professor and director of the Solomon Snyder Department of Neuroscience and the professor of biological chemistry at Johns Hopkins University School of Medicine. He's also the co-director of the Johns Hopkins Brain Science Institute. Dr. Huganir's work with the SYNGAP1 gene and its protein has evolved since 1998, when he first discovered that the SYNGAP1 gene's protein plays an important role in regulating synaptic plasticity. Since then, his lab has also created mouse models which reflect human mutations in SYNGAP1 and have characterized the resulting phenotypes. Of course, on top of all that, Dr. Huganir has been on our scientific advisory board since the very beginning and was the very first grant SRF gave. So it's a great pleasure to have him with us, sharing back some of the work that he's done, and we funded a small part of it. And we're just thrilled to have you here. So Q&A, you can wait till the end to ask your questions live, or you can also put them in the Q&A or the chat, and we will get to them at the end. With no further ado, Dr. Huganir, over to you.
So Mike, it's fantastic to be back and give another webinar to give an update on our research, and we certainly appreciate the support you've given us to start some of this, and really it's an honor to be here to present to the families, because that's why we're here, is to really help kids who have SYNGAP-related disorders. And so I think we're getting there, and I'll give you an update, sort of a summary of some of our basic science, but also some optimism about potential therapeutic approaches. Now, I'm going to start pretty basic, because there might be new families online, and so I'm going to give an introduction about SYNGAP and how the brain works. And so for those any scientists who may be watching this, I apologize for giving a very basic introduction. But so can you see that, Mike? Okay, great. So today I'm going to talk about the role of distinct functions of SYNGAP in SYNGAP-related disorder. This is a new finding we've just recently published, that SYNGAP is not only a RAS-GAP, but is we think a structural protein at the synapse, and that structure is really key for synaptic function, but also key for cognitive function, and also is a really important very relevant to how we develop therapies for the kids. So just a couple of disclosures. I'm scientific co-founder and SAB member of Neumora Therapeutics and an SAB member of Maze Therapeutics. So I'm going to start really basic. The brain is a network of 100 billion neurons that are interconnected through synapses that create circuits in the brain. And so here's just a video of neurons in the brain, animation showing how they communicate. So we have 100 billion neurons in our brain. They form millions of neuronal circuits through synaptic connections. And this is just illustrated here in this. You can see the neurons firing away, and each neuron has these elaborate dendrites and axons that contact at synapses. And we're going to zoom in on a synapse shown here. And this synaptic connection is where neurons connect with each other, signal to each other, and form these very complex neuronal circuits that underlie basically everything we do, from our emotions to our decision-making to formation of memories, basically all our behaviors. So, so my lab has for many years been interested in the synapse. And so this is just a schematic of a synaptic connection. So one of those little synaptic connections between two neurons. And this is how they communicate. They release neurotransmitters, which then diffuse across and bind to receptors on the postsynaptic membrane. And so this transduces the signal between neurons. And so neurons, these billions of neurons are firing away, trillions of times a second while you're sitting here watching this. So we're interested in this process of synaptic transmission, but we're really interested, my original interest was in learning and memory. So what happens in the brain when you learn something, and how can you remember it for a minute, 2 minutes, a decade or more? And so we've been studying sort of the biochemical and physiological changes that occurs in the brain during learning and memory. So this process is thought to be mediated by something called synaptic plasticity. And this is the change in synaptic strength between neurons. So the idea is that when you learn something, the strength of synaptic connections changes and forms a novel circuit that actually encodes the memory. And so just an example of this type of plasticity is shown here. And this is electrophysiological recordings from a slice of brain tissue in a region of the brain called hippocampus that's really critical for learning and memory. And so if we stimulate and record a synaptic response, so here we're looking at time, and here we're looking at the strength of the synapse. And you can see when you give a very high frequency stimulation, you get a potentiation. And so this is an increase in synaptic strength, and this will modify the circuits within the hippocampus to, in theory, could potentially encode the memory. So this is a process called long-term potentiation. So the potentiation is very long-lasting. In fact, in intact animals, it can last for up to a year. So this is a model of how memories can be, encoded quickly and last long-lasting. Now, there's another form of plasticity called long-term depression, which is the opposite. Basically, in the same region, you can get a nice stable baseline again. You give a low frequency stimulation, and you actually get a depression. And so these two combinations of long-term potentiation and depression help to sculpt circuits that are involved in plasticity and learning, and many other forms of higher brain function, including actually early development. And we can talk about that later as well. So many years ago, in the beginning of my career, I started to focus on the synapse. So what could be changing at the synapse? How could you change synaptic strength during this potentiation, long-term potentiation or depression during synaptic plasticity? And so we decided to focus on the receptors, how the receptors, these receptors here, are regulated. We reasoned that if you could modify receptor function, you would be modifying the strength of synaptic transmission. So we've studied a variety of ways and mechanisms using events like phosphorylation, chemical modification of the receptors to study how they are regulated. I'm not going to talk about that today. And we've shown that these receptors are highly modulated, as I'll talk about, and are really critical for the forms of plasticity across the brain, including LTP, long-term potentiation, long-term depression. So the receptors we focused on in the last couple of decades are the AMPA receptors. This is a receptor that's really mediates about 70% of synaptic transmission in the brain. So it's a really critical receptor. And we know a lot about its structure and function. And we've been studying how this receptor is regulated under a variety of circumstances. One very important thing we've discovered, along with others, is that the receptor binds to proteins, as shown here. And these receptor-associated proteins are really critical for regulating receptor function, and particularly in regulating receptor levels at synapses. So over the years, we and many others have found dozens of proteins that interact with the receptor and regulate these receptors' function. But you'll notice down here on the right, there's a protein called SYNGAP, and this is really a key protein, as we'll find out, in regulating receptor function. So, so all these proteins, what they do, many of them at least, is regulate receptor levels at synapses. So during LTP, remember, we increase synaptic transmission. So what happens during LTP, we and others over the last couple of decades have found it actually, LTP induces the increase in the number of receptors at synapses. And so this actually makes the synapse stronger and can mediate that synaptic potentiation. So in addition, the LTD, this long-term depression, is actually the opposite. What happens is receptors are endocytosed or removed from the synapse, and that makes the synapse weaker and mediates LTD. So receptor trafficking or localization at the synapse is really a key mechanism for synaptic plasticity, and really has led us to discover all these proteins and to identify that involved not only in SYNGAP-related disorders, but many other cognitive disorders such as autism and actually schizophrenia as well. So the topic for today is SYNGAP. And so, as Mike mentioned, we identified this gene in 1998, so it's been a long time. Mary Kennedy also cloned it at the same time. And so Mary and our lab and several other labs have been working on it for decades. So what we initially did is we identified this as a RAS gap. So, so the structure of the protein told us it was a RAS gap. And so it has an enzymatic domain shown here, GTPase activating domain. And this enzyme activity regulates downstream signaling through signaling pathways, which include RAS and kinase cascades and a whole pathway that's very interesting in synaptic plasticity. And so we identified this protein. It has this major GTPase activating domain, has other domains I won't get into in detail. It has many phosphorylation sites. And this C-terminal domain, which we'll talk about later, which is really critical for its function. One thing that we noticed immediately is that it's highly localized in neurons and specifically localized at synapses. So here is an image of a neuron grown in a dish. Labeled for SYNGAP, and you can see SYNGAP is really at these decoration, these little punctate decorations around the dendrite shown here, and these are synapses. So it's highly concentrated at the synapse. So back in 2003, we genetically eliminated this gene. We knocked it out in mice. And so using genetic methods back then, homologous recombination, we eliminated the gene. If you eliminate both copies of the gene, of course, we all have 2 copies of SYNGAP from our mother and father. If you eliminate both copies of the gene, the mice would die. They would die within 2 or 3 days after being born. But when we looked at when we knocked out only 1 copy of the gene, we found the mice could live, could breed. But we found, we and others found dramatic defects in synaptic plasticity, in learning and memory, and a variety of other behavioral deficits. And we also now know these mice have seizures as well. So this is some of the initial data showing deficits in plasticity. So here is LTP, as I showed you earlier. This is a wild type littermate. So this is a normal mouse. And when 1 copy of the SYNGAP gene is knocked out, you see that the amplitude of LTP is decreased. So there's a deficiency in synaptic plasticity. Now, we and others have done all kinds of behavioral tests on these mice over the decades. Just one example is shown here, and this is a really easy assay that we use routinely to sort of screen mice and screen therapeutics. And this is called a Y maze. It's measuring the working memory of these mice. So if you put a mouse in this maze, it'll run around for the first few minutes and it will explore every inch of the maze. So it'll go down one arm of the maze, then it will go here and make the choice to go down the other arm that it had not gone down before, and it will alternate and to explore the whole space. So if you have good working memory, you remember where you've been, and so the assumption is that this is a test for working memory. So if when you do this with a wild type mouse, a normal mouse, you see that the mouse will alternate about, in this case, about 70% of the time. It will go to this choice point and then go down the novel arm. And this can vary depending from 70 to 90%, depending on the wild type background. In contrast, the heterozygote, when it gets to the choice point, it basically randomly chooses. So SYNGAP mice that have only 50% of SYNGAP basically are choosing at a chance level and don't, the interpretation is they don't really remember where they've been. So there are many other tests, and we'll come back to them later. But this was just an introduction to the knockout. And so many people have made knockouts and characterized behavioral alteration or deficits in these mice. So what is SYNGAP doing? So as I showed you, SYNGAP is highly concentrated at the synapse. And so we thought, I'm not going to go through this in detail, but we thought that SYNGAP was actually playing a negative role in synaptic transmission. It's a GTPase activating protein, which actually is a negative regulator of a protein called RAS, shown here. So SYNGAP, when it's expressed very high in the synapse, will inhibit RAS function. And so we were very interested to find out how is SYNGAP regulated, and for example, during plasticity, is the activity of SYNGAP decreased so it can activate RAS, which we know from other studies is important for synaptic plasticity. And I won't, this is not, the details of this is not too important, but I'll show you the model that we generated over the years. Of course, this is built on a variety of other work from other labs as well. So here is the model, and this model is what I'm going to talk about today. Is from our recent work, this model has drastically changed. So under normal conditions, SYNGAP is inhibiting RAS, as shown here. So this is during a normal synaptic transmission, no plasticity involved. And then after plasticity is induced during LTP, what happens is SYNGAP actually physically leaves the synapse, and that allows RAS to be activated. So SYNGAP, the GTPase activity, cannot inhibit RAS anymore. And so it promotes the recruitment of more receptors and actually increases the synaptic size. And this is what mediates plasticity. So we think with in the knockout mouse with 50% of SYNGAP, this process is deficient. So this was interesting. But one thing that we and others had noticed is that SYNGAP is incredibly abundant at the synapse. So if you look at the number of copies of SYNGAP at a synapse, it's extremely high. It's the third highest expressed gene in synapses, almost stoichiometrically with PSD95, which is a major scaffolding protein that forms the structure of the synapse, but also the CAM kinase 2, another gene family that's really important for synapse structure and function. So why is a catalytic protein something that should just be present in small numbers? Why is it so concentrated? So it turns out that SYNGAP has plays a structural role, as we found out in a collaboration with Mingji Zhang's laboratory in Hong Kong University School Science and Technology University. So this is our initial paper, published in 2016. And what we found is actually if you take purified Syngap and put it in solution, shown here, or purified PSD95, that scaffolding protein I talked to you about, that's really critical for synapse structure, and you mix them together, they form these sort of what we call liquid-liquid phase transitions. So they actually form these little, almost like conceptually like an oil droplet. They come out of liquid phase, and this is a liquid droplet of these highly, highly concentrated proteins interacting together. So this was an interesting structural, pretty unique structural property of Syngap. And so we wanted to ask the question, what's important for Syngap function? Is it really the structural property that's quite unique, or is it the GTPase activating domain that's really important? So just to show these droplets are pretty cool. As again, I show you the Syngap alone, there's no formation of these liquid phase transitions. These condensates, we call them. PSD95 alone, if you mix them together, they form these droplets. And this is a video of that. When you just mix them together, you see them, the droplets form, and they actually sort of fuse together. So these have a very unique property, and many people in the field think these are really, these type of properties are really important for synapse function and structure. So how do we test this? What's important, the structure or the GTPase activating domain? So what we did is we actually mutated the GTPase activating domain. So a lot is known about these GAP proteins. We know a lot about their structure and how they work catalytically. So we could go in and surgically mutate the amino acids that are important for GAP activity. So we mutated this Syngap, these two residues shown here. And when we assayed it, this is measuring the GTPase activating protein, as shown here. This is the GAP activity of Syngap. But if we mutate those two amino acids, you see it's just like there's no GTPase activity detected at all compared to the wild type shown here. So what we did is we actually made this mutation in mice. So we mutated mice to have this GAP mutation, not to knock it out, not to eliminate the gene, but actually to mutate the gene so the gene would have the normal structure, but would not have any catalytic activity, no GTPase activity. And so when we mutated these mice, we actually saw that there was no change in Syngap expression. So here we're looking at Syngap expression shown here. And you can see that it's comparable to the wild type. Interestingly, since we're mutating the GAP domain, we would expect that the downstream signaling pathway, the RAS and ERK signaling pathway, would be affected. So we see an increase in the RAS and ERK, the downstream pathway, just like in the heterozygote. So this indicates we are mutating, eliminating the GAP activity in these mice. Now, what was immediately apparent is these mice are really different. And in fact, the heterozygotes seem to be relatively normal, but even more surprisingly, the homozygotes, so if you mutate both copies of the gene, the mice live. They don't die like the knockouts. So if you expected that the GTPase activity was critical for the functions that we observe in the mutant mice, you would expect the mice to die. And so this just shows that the homozygote lives for weeks, and in fact, they live for months, and we can breed them as homozygotes. So These mice are quite different, and they seem to be viable, which is, to me, was quite surprising, actually. So we went on to characterize these mice. Okay, so they live. So what about their plasticity and their cognitive functions? And so we compared them to the heterozygote knockout. And so here we show that this is sort of the control knockout. So here is the wild type littermate mouse. This is the normal heterozygote knockout, and you can see the deficit in LTP, as I talked about earlier. This is quantified over here. And then here is the SYNGAP heterozygote. So here we might expect the same deficit because we're mutating one copy of the SYNGAP gene, similar to the heterozygote mouse. But there's no deficit at all. You have normal LTP as shown here. Now, even more surprisingly, the double knockout, or the knockout of both alleles, so totally eliminating the GTPase activity in this mouse in SYNGAP, we saw normal LTP also. So this was really surprising to me, actually, that these mice have normal LTP even when you eliminate the GTPase activity. So what about behavior? So we went to look at activity levels, and SYNGAP heterozygotes, the classic mutation where you eliminate one copy of the gene, they are hyperactive. When you put them in an open field, they're very active, as shown here. But when you look at both the knock-in, the GTPase activating protein shown over here. So if you do a single GTPase allele or even the double gap mutation, there's very little effect on activity, and they have normal activity compared to their wild type littermates. So again, this is quite surprising. We went on to look at the Y maze, and as I showed you earlier, the heterozygote knockout has a deficit in the Y maze alternation, as I showed you in the beginning. But all of the mutations, both the heterozygote knock-in and the homozygote knock-on, have normal behavior in the Y maze. So they have apparent, this presumption is that they have normal working memory. Another property that is deficient in the SYNGAP knockout, heterozygote knockout, is cued fear conditioning. And so this is the normal wild type littermate in this set of experiments. And in the heterozygote knockout, you have no learning fear conditioning. And I'm not going to go into detail due to time today, but this is one form of learning called fear conditioning. In contrast, the heterozygote knock-in has totally normal cued fear conditioning. And there is a trend for maybe a lower level of learning in the homozygote knock-in. So we have both gap domains mutated, but there is a significant difference. There is more learning than in the heterozygote knockout. So this data, was published in Science just a few months ago, and really changed our model for how plasticity occurs. And so this new model is actually, and I'm not going to go into the detail of the model, we have a lot more data, but it says that to us that SYNGAP's role in at least these types of behaviors that we've looked at and in plasticity is really plays a structural role. And so in this case, RAS is not involved at all, and downstream signaling of RAS. And so this is the schematic. We have SYNGAP is bound to PSD95, the scaffolding protein. It turns out that AMPA receptors and the associated protein called TARPs also bind to PSD95. And what we've presented in this paper is that SYNGAP is actually blocking the binding of AMPA receptors. So once SYNGAP is bound to this PSD95. The AMP receptors can't bind. And then during LTP, what happens is SYNGAP leaves the synapse, as I mentioned earlier. And then AMP receptors can sweep in and bind to PSD95 and then increase synaptic transmission. So this is a totally different model. It depends on the structural role of SYNGAP in modulating the content of receptors at the synapse during LTP. So what does this mean? It has impact on both the basic biology, but on therapeutics. And, this is all basic biology, but why we're here today, why I'm talking to you, is because of these kids, right? And kids have mutation in SYNGAP and have SYNGAP-related disorders. So I'm sure most of you know this landmark paper from Jacques Michel's laboratory, where he found 3 children with mutations, very severe de novo mutations in SYNGAP that presumably result in haploinsufficiency. And of course, all the presentations. This produces intellectual disability. It has autistic repetitive features in many of the kids. We now know they have seizures. Most of the kids have seizures. They have sleep disturbances, and et cetera, et cetera. So this really changed the way my lab started thinking about SYNGAP. We're a basic science lab, but when we found out that SYNGAP was involved not only in mouse behavior, but also in human behavior, it took on a new urgency. And so it's been an honor really to be involved in several of the patient advocacy groups, as well as SYNGAP Research Fund. And so I'll tell you our story into trying to develop therapies for these kids. So here's just a probably outdated slide showing the number of mutations found in SYNGAP. We now know, of course, that those 3 initial children were not alone. There are thousands, tens of thousands, if not hundreds of thousands of kids across the world. There are mutations, very severe truncation mutations shown up on top, which probably almost surely result in haploinsufficiency, and I'll talk about that in a minute. That they are basically like the heterozygote knockout. We would predict they would have 50% of SYNGAP in their brains. But then, of course, there are the missense mutations down here, which are still we know little about. My sort of bias is that I think that most of these lead to proteins that are unstable, and that most of these result in haploinsufficiency too, but likely not all of them, of course. So we got involved very early, and we've been involved in both the SYNGAP1 Foundation, but also SYNGAP Research Fund. And, we've met many of the families, many of you may be in the audience at Symposium, but also, of course, we set up a clinic here at Johns Hopkins at the Kennedy Krieger Institute. So some of you have come through the clinic, met with Connie Smith-Hicks, and some of you have come over to the lab and met some of our researchers. So meeting you has really motivated me and my laboratory to really work on SYNGAP and develop therapies. It's really working at a new level of urgency. So one things we've done to contribute to the field is to generate mouse models for these of kids with SYNGAP mutations. And so we've made 2 so far that we've published and are available to the community as models for potential therapeutic approaches. In honor of patient number 3, we generated her severe truncation and just modeled it in mice. We were able, since SYNGAP is so well conserved, we were able to basically make exactly her mutation in a mouse model. And so when we analyze this mouse, again, the homozygotes die, but the heterozygotes actually show SYNGAP expression is decreased by 50%. So here we're looking at the expression of SYNGAP in wild type versus the knock-in mutation for patient number 3. And it's just exactly like the heterozygote knockout, where we completely knock out the second allele of SYNGAP. So this indicates patient number 3 is almost certainly haploinsufficient. She has 50% of SYNGAP in her brain. And when you look at the behavior, it's the behavior of this mouse is basically identical to the conventional knockout. The heterozygotes have deficits in working memory, activity, and also in some repetitive behaviors that we've looked at. So we've wanted to look at other mutations, other types of mutation, and so we also looked at a splice site mutation, and this is a mutation that occurs in the intron and near the end of SYNGAP, and it actually results in a missplicing of this region, leading to a stop codon, which we would predict would lead to haploinsufficiency. Now, this is near the 5' end, the 3' end, or the end of the SYNGAP gene. And so there was a possibility that this would not lead to nonsense-mediated decay. And so this mutation may not result in haploinsufficiency. So we generated, again, the exact same mutation in mice. And it turns out these mice are also haploinsufficient. So you can see that we analyzed different isoforms, and I'll come back to these isoforms later. There are 4 different splice variants that we look at, and you can see that all splice variants are decreased in this mutant mouse, as shown here. And also the ERK activity, the downstream RAS signaling pathway, is also increased in these mice as well. So this is all published, and in the paper you can see we've also done behavior on these mice, and they behave just like the conventional knockout heterozygotes. So again, this indicates this children with this type of mutation in cryptic splice sites are probably haploinsufficient. Now, we would like to do some missense mutations, and so we're gearing up to do some missense mutations modeling in mice, but we have not done that to date. So, summary of this part: SYNGAP is an abundant synaptic GTPase activating protein, a gap critical for plasticity, learning, and memory and cognition. LTP and learning and memory and other behaviors are SYNGAP gap independent and are dependent on SYNGAP's ability to undergo this complex structural phase transition, phase separation, or condensate formation. SYNGAP mutations in human, as we know, underlie up to 1% of intellectual disability or SYNGAP-related disorders. But really importantly is when we look at the database, humans with gap inactivating mutations do not have intellectual disability. So this is quite surprising, and we would love to, find these individuals, and they're not reported in the clinical databases. They do exist, but if we could, if they have some kind of subtle phenotypes, it would be really interesting to meet them. So now we do not think that the GTPase activity is not important. We think it's been conserved across all species. The GTPase activity is doing something. It's just not doing affecting the cognitive properties that we analyzed, or it's not important for viability. And actually, we're using these mice, the mutant mice, to try to figure out, is there something wrong with them? What is the GTPase activity actually doing? But we think this result has relevance to therapeutic approaches, and that some therapeutic approaches are looking at targeting the GTP as an activating domain, regulating the RAS signaling. And we think that if you have genes or drugs that will affect the RAS signaling, they may not be sufficient to rescue the phenotypes of both mice and humans. So I think the field, and we are, and many of us are, focusing on really increasing the expression of SYNGAP back to normal levels using a variety of approaches. So just in the last couple slides, I want to show you what we're doing to do that. There are many labs across the world, many companies that are trying to do this through a variety of approaches, and I'll mention them at the end, but we have focused on two areas. So one is development and preclinical testing of AAV, the virus expression of SYNGAP1. So this is a genetic rescue of SYNGAP1 by expressing SYNGAP1 through this AAV delivery system that many of you might be familiar with. And so initially, many people thought SYNGAP was too large for AAV expression, but Ingi Hong has really worked on this extensively and has modified AAVs to encapsulate SYNGAP1 and to get good expression. So with this, we've looked at SYNGAP expression in human iPS cells, iPSC cells. And so this is just normal iPS cell, but we can see we can actually express SYNGAP over 75-fold over normal expression in these cells. So this is obviously too much. We don't want to overexpress it that much, but it shows the viability of this approach, that we can get incredible expression of SYNGAP in human cells. We've also done this in vivo in mice. So and so here are just looking at different isoforms. So we actually we can only express one isoform, one splice variant in each AAV. So we have here alpha-1, alpha-2, beta, and gamma. We can express all of them in vivo in mice by injection, either in culture or in vivo. And so again, we can get good in vivo expression of SYNGAP. And this is one approach to try to express SYNGAP back to its normal levels in the mouse models of SYNGAP-related disorders. So for example, this just shows that SYNGAP is expressing at synapses really well. The magenta is this AAV expressed SYNGAP. The blue is a marker for dendrites, and green is the antibody for SYNGAP. So you can see nice synaptic labeling of these neurons in culture. But we also can get an expression in vivo, and so this shows the spread of the virus in mice where we've injected the virus at postnatal day 2. So what we're currently doing is taking this approach and have a pipeline where we analyze the rescue of behaviors in the mouse models of SYNGAP-related disorders, both the normal heterozygote, but also the mutant models that we've made. So we inject the virus at P0 or so, between P0 and P10, for early, postnatally, right after they're born, let them grow up, make sure they're healthy, then do open field tests. We do the Y-maze test. We do a variety of other motor tests and social interaction tests and pain thresholds. And then we do EEG to measure seizures in these mice as well. And then finally analyze the expression of SYNGAP and both at a biochemical level, but also across the brain. So we're having some initial luck with this, and so hopefully in my next update to you, I can present some positive results on this approach. Now, the other approach we're taking is using antisense oligos, and many of you also are familiar with this. We have focused mostly on what are called splice switching antisense oligos. So these are similar to antisense oligos you're used to, but we're targeting the SYNGAP gene to regulate the alternative splicing of the gene. And so many of you know that there are 4 isoforms of SYNGAP that are alternatively spliced in the C-terminal region, called alpha-1 and alpha-2, beta, and gamma. In a variety of data from our lab, but also other laboratories, we think that alpha-1 is the critical isoform, that we need to rescue alpha-1 to get rescue of behavior and plasticity. And we have some data already published on that. I'm not going to go on into it because of time. So we wanted to identify how we can modify alternative splicing to produce more of the alpha-1. And so Ingi Hong and Tracy Hannon in my laboratory did a what's called an antisense oligo walk in the intron where the around the intron where the splicing between alpha-1 and alpha-2 occurs. I'm not going to go into details of this, but we used a series of antisense oligos to look at expression of alpha-1, and you can see that some of these antisense oligos or splice switching oligos increases the expression level of alpha-1 compared to alpha-2, as shown by these different colored bars here. And in fact, when we titrate in this, our best lead ASO, you can see we can get up to a 3-fold increase in alpha-1 expression in this system. So this is a lead ASO that we are now trying to use to rescue behavior and plasticity in mice, and we're optimistic that this could be an important reagent that could eventually be used in the clinic. So coming back to my summary, what I've talked about today is really our update on the basic biology of SYNGAP, but turning the focus away from the GTPase activity and to the structural domains of structural role of SYNGAP in normal behavior, but also in SYNGAP-related disorders. And so I want to thank everybody for listening, and come back to really emphasize that this whole complex of proteins is really critical for intellectual disability, autism, and schizophrenia. So, for example, mutations in the AMPA receptor subunits that I talked about, many of you know about this, GRIA1, 2, or 3, mutations in those receptors actually result in intellectual disability as well, and rare forms of intellectual disability and autism. Mutations in the GRIA3 gene is also ultra rare mutations are really one of the top hits in schizophrenia as well. So this complex is really important in a variety of cognitive disorders. Now, funding, I'm mostly funded by NIH, but I really want to thank Mike and the research SRF for funding some of our initial work on this, specifically the generation of the mice models. I've also been consistently funded by the Simons Foundation as well. This week was a sad time for the Simons Foundation because Jim Simons died, a giant who's really changed the field of autism and autism-related disorders, sadly passed. So finally, the people involved in this. So this is the history of SYNGAP. It's a new slide, so I had a typo, but history of SYNGAP in the lab. So Ji-Hye Kim cloned it a long time ago. Hae-Young Lee did some of the physiology of the knockouts. Initially, Kogo Takamiya made the first knockout. Gavin Rumbauer was a postdoc in my laboratory. back in the early 2000s. Of course, he's gone on to be a major player in the field, really a leader in the field of SYNGAP research. And importantly, Gavin is doing, as many of you know, a chemical screen to look at, not to regulate the GTPase activity of the protein per se, but to look at compounds, small molecules that will regulate the expression of SYNGAP. Tim Gamache was involved in some of this work, as well as Sarah Jew and Casey Rekovich. And as I mentioned, our initial collaborations on the structure and function of SYNGAP were done in collaboration with Ming Chi's laboratory at Hong Kong. This is the SYNGAP team. Yoichi and Ingi have been the pioneers in this work and really are amazing scientists. But we have a broader team of Blaise Clark, Elizabeth Gerber, Tracy Han, who's done this beautiful work on splicing, Sion Lee, and Hai Tran as well. So this is the lab right after COVID, our first gathering in my backyard. And a great group of people. And I always say that I do not employ children or dogs in the laboratory, but they were present at the party. So I'd be happy to take any questions if there's time and interest.
Thanks, Rick. It's a great presentation. There's a couple questions in the chat, but before we get to them, can you talk a little bit more about isoforms? I mean, this is something that has been flagged as an issue or not an issue. And you basically, if I heard you correctly, you said that alpha-1 is what that ASO is focusing on. So the working as the, when you and others have said this, the alpha-1 isoform is the one that people tend to focus on. But there's that, just as the RAS gap is, you know, it still serves an important function. Those other isoforms serve an important function, right? So one of the questions that comes up is, if we had a therapeutic approach that favored one isoform over the others, we are assuming, and we have shown in other papers, that that isoform really helps rescue certain functions. But we are then implicitly accepting, this is the question, are we then implicitly accepting that the other isoforms we're just going to do without and be happy that we're been hope / assume that being haploinsufficient in those other isoforms is okay? Is that?
So yeah, we don't know why, but so in some of our early work in vitro, and this is published as well, so we knock down SYNGAP, and then we rescue with various isoforms. And at least in our plasticity paradigms, the alpha-1 is the one that rescues the best. It's really very efficient. Now, interestingly, the beta isoform is really important for dendrite development in our assays as well as in other assays. But I think a key finding there is that, you know, and this goes back to Gavin's earlier work, where he was able to rescue in a relatively adult mice, you know, the some of the behaviors by reintroducing SYNGAP. So he showed that if mice, you know, the heterozygotes are, you know, if you express after 3 months, you know, 3 months, you express more SYNGAP, you can rescue a lot of the behaviors. So so that suggests if there's any impact on early development, this is to address another issue about SYNGAP during development. Even if the early development you have a haploinsufficiency, you can live through that and recover behavior. So so we think that if there are dendritic deficits in say in even if you overexpress SYNGAP, for example, that the mouse or the human could live through that early, maybe a delay in the dendritic development, and then have normal function afterwards. So it's really unknown, you know, what would happen. Now, one advantage is the AAV approach. We have all the isoforms, right? So we're going to be testing all the isoforms alone and in mixtures. So in theory, we can try to rescue with just alpha-1, see if we get rescue, or you try the others, or even mixing them in the right proportions and expressing them in mice. So that will address one issue. But there's some other sort of esoteric experiments that we have done, and also some nice work from Gavin's laboratory suggesting, again, that the alpha-1 is really critical for not normal cognition. So in experiments we haven't published yet, but based on the splicing work, we made new mice with again surgical mutations in splicing to eliminate alpha-1, only alpha-1, and those mice are like heterozygotes, knockouts. So if you mutate just alpha-1 expression, the mice have deficits and are sensitive to epilepsy, have epilepsy. So we're encouraged that just increasing alpha-1 will have an impact. It doesn't mean the other isoforms are not important, but maybe if you have 50% of the other isoforms, that's sufficient for their function.
Right. And then just I'm curious to hear your answer to this. I've heard people say there's 4 isoforms focusing on the 3 prime. Is it the 3 prime? The N?
Yeah.
Right. But there's also 3 at the 5 prime. And then some people are going like 3 times 4 is 12. How do I describe SYNGAP's number of isoforms? Do I just say it's 3 on this end and 4 on that end, or is it? I mean, how complicated is this?
As you say, their permutation combinations are 3 times 4. So the start sites are interesting, and we don't really understand it. At least I don't understand. The critical role of the 3 prime, the start sites. But if you look at the genetics, mutations, as you know, up in the beginning of the SYNGAP A isoform are pretty low. So mutations within that domain probably do not produce intellectual disability at high frequency. And so we think that if we just express the beta isoform, that will get rescued. So we are focusing on the beta isoform within the AAV studies, and so I think we're focusing more on the C-terminal regions. We think those are more critical for function.
Got it. I have a million questions about isoforms and the different functions. I mean, one of the things we see, and it's in the Vlaskamp paper, is mutations in the first 4 exons sometimes result in a much milder phenotype, right? And I think the working assumption there is that's just because some of those, depending on the start point of the different 5 prime isoforms, some of them just get skipped, and so patients have probably more than 50%, but less than 100. So then the big question in the Q&A, and I would urge people, if people are desperate to talk to Rick, raise a hand or send me a message and I can add you to the panel, but otherwise I'll just read out of the Q&A. Aaron was just very excited in this webinar and has 3 questions. I want to emphasize his point about we're excited that you're looking at missense. I see on this call Michael Courtney is here, and as you know, we're funding some good work in Finland around looking at missense both in vitro and in silico. But Aaron asks right out the door, is SYNGAP a RASopathy? Because this is a question that comes up. It is the basis for SYNGAP's inclusion in a big study in Europe right now. RASopathy for some people means tumors, so this has always been a point of concern for families. Given everything you just talked about with the gap domain, how do you answer that question?
So, it has a RAS, GTPase activity domain, and it regulates RAS, and so I think that's going to be playing some role in but I wouldn't call it a classic RASopathy because of the structural role. And so I don't think I think studies looking at the RAS activity and the GTPase activity are really important and can contribute. But I think to get full rescue, we just can't rescue the GTPase activity domain or GTPase signaling. We need to increase SYNGAP to play its role as a structural protein in the synapse. So so that would be my answer. Probably is a little controversial at this point, but we can't just look at one or the other. We need to do, look at both.
Yeah, we need a T-shirt. SYNGAP. It's complicated, right?
It's actually really interesting protein, actually, at a basic science level.
That's good. I mean, it's held your attention for 26 years from 1998 to today.
Yeah.
You've been working on this. It's incredible. So Aaron has another question, which I'm going to let you read the second one because I'm actually, can you see the Q&A?
Can't see.
I don't want to misspeak it. But while you're looking at that, Heather asked two questions around ID.
Mm-hmm.
And is sort of her last one is the classic question parents ask: given your experience learning memory, is there a period of development by which you'd have to institute these therapeutic approaches to have a positive effect in humans? Said different, when is it too late to intervene?
Yeah, so I think certainly the goal is to intervene as early as possible, right? So and I think the key to that, as you know, and the parents know, is early diagnosis and so I think getting any kind of reagent, antisense, AAV, small molecule into the kids early is going to be really key. And I think there is a window. We don't know what the window is. I mean, I think Gavin's work says you can get rescue of some phenotypes relatively late in mouse development. So whether that's true in humans. So I think there's optimism up to a certain age, but there's definitely going to be a window of opportunity. So we need to get these things into kids, as soon as possible. And I'm optimistic about that. So let's see what else.
Oh, I have two more while you're digesting. So both the therapeutic approaches you presented sort of favor an isoform. What about other modalities that might just, I mean, if Stokes Tango is public, right? Mm-hmm. Just make the wild type make more and let the body worry about which isoform to express. Mm-hmm. I mean, it just at a theoretical level, like given the complexity of isoforms and all the things we don't know, those are sort of elegant and attractive from that perspective. Do you agree, or like why?
No, totally agree. I mean, I think that would be the best approach. If we can find reagents, not the obvious choices, antisense oligos to impact splicing or other regulatory expression of the gene, that would be the best. If you can upregulate all isoforms equally in their correct proportions, that would be the best approach. Now we have not had much luck, and that's something we've been trying to do through a variety of techniques, as you know, and we haven't found something that really raises the expression level of SYNGAP well enough, especially later in development, to justify pursuing it further. I hope Stoke and other companies have found better results, but so that's why we've turned to the alternative splicing. The alternative splicing is really interesting in a basic science level, because the alpha-1 is totally non-canonical splicing. It should not be there according to most people who study alternative splicing, and so that's what Inge and Tracy they've really mapped that out and characterized this really bizarre alternative splicing. And targeting that's going to be incredibly specific. There's only one other gene in the whole genome that is spliced in a similar manner. So if we can target that form of splicing, that will be a very specific drug. That's another advantage in looking at the alpha-1 splicing.
Interesting. Okay. So Aaron's question was, with the knockout of the GAP domain, this would emphasize functional significance of the other domains, especially C2, same for pH. Is there an interest in looking at other domains and their role? What domains bind to PSD95?
Yeah, so we've been doing that, and it's in the paper, sort of. So if you mutate the regions that interact with PSD95, that disrupts, has an impact on this liquid phase transition, these bizarre structural effects, and that does not rescue plasticity. So we are trying to make, for example, we're trying to make a mouse where instead of mutating the GAP domain, we have a surgical mutation, but that will only affect that liquid phase transition, and then see, is that mouse normal or is that mouse have deficits? And so we like to make very surgical, simple mutations, not large mutations. And so we haven't found that sweet spot for to knock out the liquid phase transition.
Got it. So I have one question. I was at a conference last week, the Milken conference, talking to people about SYNGAP, and this week I'm at the boot camp of Ultragenics, and I talked to I don't know how many people about SYNGAP in the past 10 days. And one of the questions that always comes up when you say upregulating is, what does gain of function look like in SYNGAP? And my answer is, you don't see gain of function. And they're like, why not? Is it lethal or is it just not a big deal? And then we somehow tau always comes in as, well, tau regulates SYNGAP and maybe there's natural regulation. And what I heard you say, if I got it right on my last question, was when we try to upregulate across the board, somehow it doesn't work. And so that's why we've gone for a specific isoform. So the sort of two questions thrown together. One is, what is your thinking on, could any of these potential, they're all theoretical, right? We don't have them in human yet. We're just in mice. What happens when you upregulate SYNGAP so much that you go over 100%? And then is that a big deal? Is that something we should really worry about? And then the related question is, could the body's regulation of SYNGAP be one of the reasons those other therapies are not proving out in the lab?
No, so let me clarify that. So at least the approaches we've taken, the ASO approaches we've taken to directly try to increase splicing of all isoforms have not worked in our hands. So,
right.
So that's what I'm saying. It's not that they, increase it so much that it's a problem. Now with the AAV, of course, overexpression is an issue, and that's a concern. And of course, all AAV genetic rescue approaches. So really, nobody knows what overexpression of SYNGAP does. I mean, when you look at the human population, the expression of SYNGAP is very tightly controlled, and the isoform ratios are very tightly controlled across all individuals. Inge has found, I think, one person who had like 50% increase of RNA, SYNGAP RNA, but we don't know, we don't know who that person is, and it's not in a clinical database. But one of the side, results we'll get from the AAV experiments is we will highly overexpress SYNGAP in mice before we titrate down to get, just a twofold effect. And at least preliminary data, when we overexpress SYNGAP in mice, they don't die. There's no obvious distinctive behavior, but we haven't looked at subtle phenotypes and plasticity and things like that. Are they smarter? I don't, who knows? But my guess is overexpression is not going to be good. Similar to Rett syndrome, that overexpressing in that case also is detrimental, as you know.
Interesting. Other questions? Everybody, I'm thrilled with the attendance, and I'm sure this will be a much watched webinar. But are there, before I thank Professor Huganir for his time and go back to this conference, is there anyone else with questions or comments? Stun them into silence.
So I'm going to say that, you know, 20, however many years ago, there was only a few labs working on SYNGAP, and it's really exciting to see so many labs joining, especially young scientists who are joining and working on SYNGAP. You know, it's probably up to 50 labs now, I think, are working on SYNGAP. And also, it's really great, the interest in biotech. I mean, we are having a lot of interest in the biotech. We know several labs that are, several biotechs that have, you know, publicly said they're working on it, but I know you, Mike, you and I know there's a dozen or more of biotech and pharma that are really interested in SYNGAP. So I think it's exciting time, and you know, I'm optimistic.
Yeah, I think when I started this, I was worried about the science. I'm no longer worried about the science. I think with the work you've done and others and all these labs and the not just young labs, but young labs led by really brilliant people, who in some cases SRF has recruited. I'm very proud of going knocking on Juliette Knoll's door and saying, why aren't you working on SYNGAP, and convincing her to do that. So, you know, I think the science is good. I think the things that scare me now are the clinical. I'm worried about what we're going to measure given the phenotypic spectrum and the challenge in what to measure, right? And then I'm also worried about the regulatory. That might shift depending on when we come in, but there's a lot of uncertainty for all these biotechs. When the biotechs call me, they're like, hey, here's the science, yay. Let's talk about regulatory and clinical trial design. And I think that is where SRF is focusing our energy and our time right now, both in looking at biomarkers and endpoints, thinking through regulatory strategy. And also, frankly, I'm thrilled to see so many families on this call, because hopefully in the not too distant future, parents will have to get diagnosed, get over the grief, learn a lot of science really fast, and then start choosing, if we're so lucky, between an AAV9 and an ASO, and then wonder about what does one and done mean for AAV9, and even if there's better AAVs, what kind of reactivity could there be if I use this now to what happens later, and then they have to start thinking about this ASO versus that ASO. I mean, the future is not simple, and one of the things we think about a lot is how to start socializing those questions, because I think they're coming sooner than they're not coming soon enough for us, but I think they're coming sooner than some people think.
Yeah.
You know?
And I think, you know, the community is ready, I think. And I think it's important that community comes together to talk about these issues. And, you know, there are thousands of these kits, but we only have identified, you know, whatever the number is, 1,200, 1,400 now. And, you know, and we need as many people as possible for each of those clinical trials. So we need to get agreement among the community, you know, what are the best bets for a therapeutic approach.
Right. And that will, and it's just really interesting to watch what people who are half step ahead of us are doing, like Angelman, Rett, SCN2A, CM1A. You start to see all these different trial designs. I can't remember off the top of my head. I've said this before. There was one company who did a trial sort of 2 to 7 in the US and then 8 to 15 in the UK, right? Just really thinking hard about who at what age will be able to do what. Because I take your point on, you know, and so we are, of course, connecting with and working with every family who wants to talk to us, and also making sure that, I mean, we're all putting kids to bed at night who are getting bigger, stronger. And when they get dysregulated because they outgrow their meds or because they're on the wrong med or because they had a bad day or whatever, people pay the price. And so there's a lived reality of families where we also have to, there's an urgency that is just very real.
Yeah.
As we think through those things.
Mhm.
Anyway, thank you. It's great to have you back.
Good to
And
see you.
keep going. And all those families sign up for Citizen and make sure SRF knows where you are. So when it is trial time, we can let you know.
And if you come through, Hopkins, go to Kennedy Krieger and see, Connie. Let me know and come over to the lab. We'd be happy to meet the families.
That'd
Show
be
'em-
great.
Show them what we're doing.
Careful what you wish for. Could have an avalanche. Thanks again, Rick.
All righty, see ya.
Bye-bye. Thanks, everyone.